This is the DNA Replication Lecture. Before you start this lecture, you should know the answers to these basic questions. Please think about it for one moment and make sure that you understand each of these topics. When in the cell cycle does DNA replicate? If something has the suffix "ase," what does that mean? If there's two bonds in DNA, which one is the easier to break? And what is meant by the term complementary? Pause the video now and make sure that you know the answers to each of these questions. In the cell cycle, DNA replicates during the S stage. That happens after G1, but before G2. If something ends in the suffix "ase," it means that it's an enzyme. It performs some type of reaction within a cell or allows a reaction to occur. Of the two bonds in DNA, that would be covalent bonds and hydrogen bonds. Hydrogen bonds are by far easier to break. Those are the bonds that hold the two strands of the DNA together in the middle, whereas covalent bonds are holding together the phosphates, sugars, and bases. The term complementary means that they match up. They're not exactly the same, but they work well together. Complementary bases are A and T, and C and G are also complementary. DNA replication is the steps in order to make more DNA. We're making an exact copy of the DNA that already exists so that when a cell divides, each cell has its own set of DNA. Here are the steps in order to do that. First, you unwind the double helix. This is performed by the enzyme helicase. I remember that because helix and helicase start with the same prefix, so it's unwinding that double helix. Second, the open strands are stabilized by proteins called histones. It's important for this to happen so that they don't get tangled or bent or broken because the DNA is very precious to the cell. Third, a copy of the DNA is made by adding complementary nucleotides to each side of the DNA. This is done by the enzyme DNA polymerase. Since we have two sides to the DNA strand, that means that we're going to have two sets of DNA once this is finished copying. Fourth, we check for errors. There's an enzyme called ligase that performs this function to make sure that we're not getting very many mistakes. Sometimes mistakes will slip through, and that's called a mutation, but we try and minimize them by having a proofreader, just like you would with an essay. Each DNA molecule will then consist of half old and half new DNA. This is called semi-conservative. Semi because it's partially, part is new and part is old, and it's conserving some of that DNA, using the old stuff to make the new part as well. The charges on the DNA cause one side to be leading and one side to be lagging. You don't need to know the details of this, but it's just good to understand for when you see the videos and the pictures that are coming up. So here's a picture of the semi-conservative DNA replication. This would be our old parent strand. And there's helicase that unwinds the DNA and separates it. So then we have half here and half here. The DNA polymerase is adding new bases and pairing them up with their complementary base so that we have one new strand that is half old, that's the yellow, and half new, which is the blue. This goes in opposite directions. This leading strand here is going straight in toward the helicase, and it just moves continuously. On the other side, the lagging strand, the polymerase has to move away from the helicase. So it causes these little fragments, and then it loops back and goes some more and loops back as the helicase unwinds the DNA. Ligase, here it is doing some proofreading and also connecting the strands together. Here's another picture of the same thing. So here is the parental strand. Here is the helicase that's unwinding and separating the DNA. So we have the old strand here and the old strand here. The DNA polymerase is adding bases here on the leading strand. It's just going straight through. And the polymerase is also moving in this direction on the lagging strand and then looping back to add more. So it comes in fragments. Again, you don't need to know the details of why it's leading or lagging or what an Okazaki fragment is, but when you see it on the videos, it's good to know why it's like that. In Blackboard, I've provided 2 links to videos that are animations of this process for you to supplement this lecture. Please contact me through a message if you have any questions. Thank you.